Okay. So, in spectro and UV spectroscopy, basically we measure the absorbance of a particular analyte at its lambda max, as Sir has told. So, in this instrument, we use UV wavelength as well as visible light spectrum. So, here what we do is, let me take the highlight. Yeah. So, talking about the instrumentation part of the device, here in this device, we have a light source which emits light at different wavelengths. So this light passes through a wavelength selector, which acts like a filter. Through this filter, only the light of particular wavelength uh, passes through because we require the light only at this wavelength because the analyte which we are going to analyze absorbs light only at that wavelength, and this wavelength is called lambda max for that analyte. So therefore, this wavelength selector has the job to reduce all the light to a single wavelength. And when this monochromatic light, the light of the single wavelength, passes through this sample, some amount of light is absorbed by the analyte and rest amount of light is transmitted further. The light that is transmitted further is detected both in its wavelength as well as in its intensity by a detector. And this detector converts these optical signals of the light into electrical signals. And these electrical signals are presented to us in the form of output. So this is the basic design of a UV spectrophotometer. So as we know, uh, whenever the light incidents are sampled, so our sample will be present in a thing like this that's called qubit. So when the light passes through this sample, the analyte that is present in our sample will absorb some amount of its light. And UV spectrometer does the job of giving us as to how much absorbance has taken place. Based on this absorbance, we determine whether the sample or the analyte is present or no. And if it is present, then what is the concentration of the analyte? And how do we do this? We use a simple law called Beer-Lambert's law. So Beer-Lambert's law simply defines an equation, which is a relationship between the amount of absorbance and then the concentration of the analyte in the sample. So according to the Beer-Lambert's law, the absorbance, according to this law, the absorbance of the sample is directly proportional to the concentration of analyte in the sample. So greater is the concentration of analyte, that is, greater is the analyte particles. So greater will be the absorbance for the analyte. And similarly, here L represents the length of path that the light travels through the qubit. That is, you can see here, this is the qubit. Through this qubit, the light passes. The length of this width, this part, this represent L. So greater is the L, greater is the length of solution through which the light passes, greater will be the absorbance. So therefore, the amount of absorbance is also directly proportional to this path length. So here, according to Beer-Lambert's law, I will summarize. The absorbance is directly proportional to both path length and the concentration of the analyte. And epsilon represents a proportionality constant. Epsilon is called molar extension coefficient. So epsilon has the units, as you can see here, liter per mole per centimeter. And if we apply these units in this equation, liter per mole per centimeter, uh, with the units of light, so we will see that absorbance has no units of itself because the units of epsilon are cancelled with the units of concentration and path length, that is mole per liter and centimeter. So mole per liter of concentration and centimeter of path length are cancelled with the units of molar extension coefficient, that is liter per mole per centimeter. So therefore, absorbance has no units of it. And secondly,
Sir,
if we, yeah,
Sir, but does the Muller extinction coefficient exist if we don't have the law like B. Lambert's law, A is equal to epsilon LC?
So
Like,
can you reframe your question?
do you have any other application of this law except using in this equation? Uh, uh, do you have any other application of molar extinction coefficient except using
Okay.
in this equation?
So Molar extinction coefficient is just a proportionality constant which is used in this equation only.
Okay, so
Okay.
okay, sir.
Yeah.
Okay, so then why are we saying that A has no limits, no units because of the units of epsilon and L and C?
What I'm saying is absorbance has its no unit. Absorbance doesn't have units because the units of epsilon cancels out the unit of the product of concentration and path length. And why absorbance doesn't have a unit, I will explain later. So as we can see here, absorbance is defined as the log 10 of I naught upon I. So I naught represents, as you can see here, the incident light, the light that is emitted onto the cuvette. This is I naught, the incident light. And I is the transmitted light, the light that gets transmitted after passing through the cuvette solution. So the ratio of I naught upon I, the intensity of incident light divided by the intensity of transmitted light and its logarithm of base 10 gives us absorbance. Okay, so this ratio I naught upon my I is 1 by transmittance. Transmittance is the fraction of light that is transmitted. So basically, absorbance is nothing but a log 10 of a ratio. Therefore, it doesn't have a unit. So these are the two reasons why absorbance is a dimensionless quantity. Okay. So this is the Beer-Lambert's law. Using this law, we will be determining the concentration of our sample. How? So doing our experiments, we will see the absorbance of our sample using the spectrophotometer. Epsilon value are always constant. L values will be the length of the cuvette. Sorry. Uh, L value will be the length of cuvette and C will be the concentration which we will determine. So by using absorbance, epsilon and L, we will determine the unknown C. And how we will determine it will be explained in the protocol of the experiment. So let us move on to the advantages and limitations of UV spectroscopy. The principal strength of this technique is that it is non-destructive. That is, uh, the sample that we use in the cuvette, we can reuse that sample. Uh, the incident of UV light or visible light does not disrupt the composition of the sample, or it does not corrode the sample. We can put the sample in the cuvette, analyze it, and reuse it for any of our application. So the first advantage is that the technique is non-destructive. It does not corrode the samples. The second advantage is it is quick. The analysis time is from 2 to 3 seconds. We can get a result in a fraction of time. So it is very fast to get the absorbance of our given sample. The third strength is that it is very easy to use. It does not require any expertise or training, specialized training with the instrument. So it is very easy to use, and anyone the basic knowledge of how to put the sample and how to take it out and how to analyze the reading, the person can use it easily. And the second thing is once we get the data from the device, it is very easy to analyze this data, and we can convert it into graphs, and that we will see in the experimental parts. The last strength of this instrument is that it is inexpensive and hence it is easily available in different labs. So these are the strength of UV spectroscopy. Now, talking about its limitation, the one primary limitation of UV spectroscopy is the effect of stray light. So when we use a cuvette and we put it in the chamber in the spectroscopy, as we will see, there are chances that any stray light will also incident on our sample, and that will change the reading because uh, the whole principle of spectroscopy is based on incident of light on the sample, as I've already told. So if we have a stray light that also incidences on our sample, it can alter our reading because we are not taking into account the extra light, extra stray light. Okay. So this is the primary disadvantage of the sample. Therefore, we require the cuvette to be completely enclosed in the chamber before we take the reading to not allow any kind of stray light to enter the sample. The next disadvantage is the scattering of light. So we have seen that inside the cuvette, the light is really more clear from this. Uh, when the light is incident on the sample, uh, some part of light is absorbed and some part of light is transmitted. Is there any third possibility that can happen to the light? Can you imagine anything else that can happen other than absorbance and transmittance? As we can see, when the light is incidented on the sample, some part of it is absorbed, while the rest part is transmitted. However, we see that sometimes a fraction of light is also scattered, and this scattered fraction of light is not taken into account in the transmitted form, neither into the transmitted portion and nor in the absorbed portion. And this results in alteration in our measurements because this is the light that is lost uh, from our detector. We do not detect this scattered light. Therefore, it always causes alteration in our measurement. So this is another limitation of this device. So that is light scattering. The amount of light that is scattered from our sample uh, is not measured, and it brings various errors in our measurement. So therefore, it results in irreproducible results. So the next weakness is the interference from different absorbing species, because in our sample, our analyte is not the only species or only chemical species that absorbs the light. There can be many more things that are called impurities. So therefore, while preparing our sample for UV spectroscopy, we need to be very careful that no kind of impurities, which also has the same lambda max as our analyte, enters the sample. So we have to minimize all these kind of impurities to prevent the interference. Now the next weakness is geometrical concentration, in which if we put the cuvette in a wrong position, or if we wrong position, or any of the components of the instrument are not aligned in its proper place, then it may lead to erroneous measurements. So the position of the cuvette and the and other components is very important. So these are the weaknesses of UV spectrometer, which can be easily avoided with careful handling. Now, applications of this technique, we see that DNA and RNA, which are very common analytes that we use for UV spectroscopy, they show absorbance, maximum absorbance at the wavelength of 260 nanometer. So this wavelength becomes the lambda max for DNA and RNA. So whenever we have a solution of DNA or RNA, we take the absorbance and lambda max. We can determine the concentration of DNA or RNA in the sample by using the absorbance at 260 nanometer. Similarly, for proteins, we use absorbance at 280 nanometer and 230 nanometer. And what is the thing that these analytes contain that cause this absorbance? As you are very familiar of, we have noticed that all the compounds which have a benzene ring like structure, they have electron delocalization property in them because of this resonance. So all these compounds which contain benzene ring-like structure, they absorb light. They absorb light. And so because of this, uh, we might know that proteins contain some amino acid which have these benzene rings, like tyrosine, tryptophan, and also phenylalanine. So these are the three amino acids because of which proteins absorb light, and they absorb light at the wavelength of 280 nanometer. Similarly, nucleic acids like DNA and RNA, they possess these nitrogen bases like adenine, guanine, cytosine, thymine, or uracil. All these nitrogen bases contains these benzene rings. Due to these benzene rings, they absorb light. So these are the reasons why these biomolecules absorb light at their respective lambda max. Now, what are the application? What are the other application than determining their concentration? So, when we take absorbance ratio at 260 by 280, like we take the absorbance at first at 260 nanometer and then at 280 nanometer and find out the ratio of both the absorbance. This gives us the purity of our samples. That is, since we know 280 nanometer means DNA or RNA, and 260 means DNA and RNA, and 280 means protein. So greater is this ratio, greater will be the DNA and RNA concentration in our samples. So this gives us the purity of DNA or RNA in our samples. So if this ratio comes out to be around 1.8, then it means that the given sample is pure in DNA. And if this comes out to be around 2, it, it, it is taken as a reference that it will be pure for RNA. Similarly, we also use the ratio of 260 upon 230, which indicates the purity of RNA and DNA samples by 2.15 and 2.50. So these are the applications of UV spectroscopy that we can determine the presence of a sample by noting its absorbance at its lambda max. We can determine the concentration of the sample, and also we can determine the purity of our sample, whether it is pure in DNA or RNA or even for protein. So, and also another application is that we use UV spectrometer at 600 nanometer for determining the concentration of bacteria in our sample. So, whenever there is bacteria in our sample, whenever there is bacteria and we put the light of 600 nanometer to our sample, some a major part of light is scattered away because bacteria are very large in size, and 600 nanometer wavelength light is also of large wavelength. So this light is scattered away, and only a small portion of light is transmitted. We measure this light, we detect this transmitted light, and based on this amount of light that is transmitted, we can determine the concentration of bacteria that is present in our sample. So this is another application of UV spectroscopy, in which we can also determine the bacterial concentration. So here, there was the application, and now we will move on to the practical component, in which we will determine the protein concentration using Foley-Lorry method. So I will hand this over to Kritika ma'am, who will practical part.
Hello everyone. The application part of this experiment, this UV visible spectrophotometer, is the colorimetric estimation of proteins. So the reagent we are going to use is a Pauling-Lowry reagent. So this am I audible? Okay. This reaction consists of two parts, Biuret reaction and Lowry's reaction. So the Biuret reaction consists of protein binding with copper ions. Basically, the peptide bonds in the protein reduces the copper ions to cuprous ions. Next, this protein copper complex reacts with Pauling reagent and forms a blue colored complex, which gives absorbance at 650 to 700 nanometers. Now, the phenolic groups present in the protein, mostly tyrosine and tryptophan, react with the following reagent and give blue-purple color. And this reaction is catalyzed by copper and this reaction is catalyzed by cuprous ions, which are oxidized, which are reduced by the peptide bonds in protein. Next, the blue-purple color is majorly given by tryptophan and tyrosine, and this reaction is pH dependent, and it works best in alkaline conditions. Now, there are few advantages and disadvantages associated with this technique. It is actually 10 to 20 times, 10 to 20 times 10 to 20 times more sensitive as compared to
Ma'am, we cannot hear you.
so it is 10 to 20 times more sensitive as compared to UV absorption at 280 nanometer. As Shubham has told you before, that you can estimate the protein quantity by directly measuring absorbance at 280 nanometer. But this technique, which involves the following reagent, it is 10 to 20 times more sensitive. This means that it can detect the protein concentration at much less concentration as compared to direct taking absorbance at 280 nanometer. And also it is more specific and less interrupted by turbidity. And it is more sensitive than other methods like Ninhydrian reaction and Biuret reaction. And it is simple to perform and can be used in small scale labs. Next, and its disadvantages are uh, the amount of color that develops differs from protein to protein. Uh, uh, so the reason behind this is that the protein, the amount of tryptophan and tyrosine differs from one protein to another protein. So the absorbance generally varies from protein to protein. So only you need a standard curve for that protein before you take absorbance of unknown. Next, it is less constant than Biuret reaction. Biuret reaction basically involves the reaction of peptide bonds with copper ions, and blue purple color is formed within that reaction. It is less constant because here in the Biuret reaction, peptide bond is reacting, but in this Pauling reagent, tyrosine and tryptophan are reacting, and they vary from one protein to another protein. but it is more constant than taking absorbance at 280 nanometers, because in this reaction it is generally combined effect of both Biuret and Laurie's reaction. So it is more constant than taking absorbance at 280 nanometers. And also color is not exactly proportional to concentration sometimes. All of these reasons are because takes it actually takes into account the content of tyrosine and tryptophan. Next is the applications of this technique. It is used in measurement of protein during enzyme fractionations and mixed tissue proteins. So if you want to do isolate enzyme from a cell culture, bacterial cells or other type of microorganism, you need to quantify the amount of protein that you have extracted. And also to calculate enzyme activity, you need to know the protein concentration. So this is one of the method which you can use. And uh, it can measure the amount of protein at a very low concentration. Above 5 mg per ml, you can estimate. Next is, we can estimate the protein in highly diluted samples also. Now the material that are required for this experiment are solution 1, which consists of sodium carbonate, solution 2, copper sulfate and sodium potassium tartrate. Now we have to mix these solution 1 and solution 2 in 50:1 ratio to prepare an alkaline solution. And then we have a Folin reagent. This is actually diluted into half by adding water. And then we have standard protein, 1 mg per ml of BSA, bovine serum albumin. Now the method that we use in this experiment is we add 2.5 ml of alkaline solution to 0.5 ml of test solution. Test solution we prepare by adding protein and uh, distilled water in appropriate proportion. Next, after adding alkaline solution 2.5 ml, we allow the mixture to stand for 10 minutes, and then we add 0.25 ml Folin reagent, and after mixing it, we keep it for 30 minutes, and then we take absorbance at 750 nanometer, and after taking absorbance, we have to plot a standard curve, either using Excel or graph paper that I will explain you later. And through this, we can get the concentration of an unknown solution. These are the reagents required. First, solution 1, which is made of sodium carbonate and sodium hydroxide. Then, solution 2, which is copper sulfate and sodium potassium tartrate. Before the experiment, these two solutions these are the reagents required. First, solution 1, which is made of sodium carbonate and sodium hydroxide. Then solution 2, which is copper sulfate and sodium potassium tartrate. Before the experiment, these two solutions needs to be mixed in appropriate proportion. Next, Follin's reagent, which is diluted with half of the volume of water. and then BSA stock solution 1 mg per ml. And these are the clean test tubes required. So we'll start with preparing solution, alkaline solution. So we'll mix solution 1 and solution 2 in, we'll take 1 ml of solution 2, which is containing copper sulfate and sodium potassium tartrate, and add into solution 1. We'll mix this solution. As you can see, this dilution table. First, we have given different protein concentrations and with a blank and a test. So, using these different concentrations, we'll prepare a standard graph and will determine the concentration of unknown sample. So, how we will prepare different standards is by taking different volume of protein solution, that is 1 mg per ml stock that we have, and add corresponding volume of distilled water, followed by addition of alkaline solution that we have just prepared, and then we will add Follin's reagent. So we will start with first tube, which contains 0.1 mg per ml of protein solution. So we'll add 50 microliter of protein and 450 microliter of distilled water. We'll set the volume of the autopipette to 50 microliter, and we will add BSA. We will add 450 microliter of distilled water. We have to set the autopipette back to its highest value before, after every use. Next, we will add distilled water. After this, next we will add 450 microliter of distilled water. Next we will add alkaline solution 2.5 ml. After making the solution, we have to vortex the tube so that all the contents are mixed properly. This is vortex. Like this, the tube is mixed properly. After mixing properly, we have to allow the tube to stand for 10 minutes or longer, and next we will prepare all the tubes according to the dilution table. Like the first tube, we prepared different concentrations of protein and mixed them properly. After 10 minutes incubation, we will add Collins reagent that I have diluted 2 times with molecular water. These are the Collins reagent that I have diluted 2 times with molecular water. After addition of Collins reagent, we will vortex. We will keep the tubes in dark for 30 minutes. Similarly, all the tubes were prepared and Collins reagent was added and vortexed. After 30 minutes, you can see the change in the color of the samples. With the increase in concentration, the color intensity has also increased. Now we will take the reading on UV visible spectrophotometer. So this is a UV visible spectrophotometer. First step is to set the wavelength at which we have to measure the samples. Measure the single wavelength, enter. So here the wavelength is already set to 750 nanometer. Next, now this is cuvette. In this, only 1 ml is generally preferred to be added, and as you can see, it has a path length of 10 mm, which was described in the protocol. So first we will wash the cuvette with distilled water. After this, we will add blank solution into the cuvette. Now, one thing is to avoid touching this transparent side of the cuvette, because if we touch, the dirt gets attached to the transparent side and it obstructs the light path. So we have to keep the transparent part in the part of light. So it's better to hold the cuvette from the top side and keep the cuvette in the path of light. Now we will set the blank in the instrument. Here the absorbance is 0. Next, to wash the cuvette after every use, but since it is blank, we can use the protein sample just after this without washing. So this is our first concentration of protein sample. We will wipe the corners, kept the cuvette, and we will press sample. So the absorbance is 0.160. Similarly, we will calculate the absorbance of all the other protein samples as well as the unknown. After this, we will plot a standard graph using these absorbance values. So this is the dilution table that I showed you in the video. So here you can see the absorbance that is measured using UV visible spectrophotometer. Has anybody any problem? Face any problem while understanding that video? And anything?
I have a question.
Yes, please ask.
Ma'am, did you added Pauline's reagent in the blank sample also?
Can you please repeat the question?
I am saying, did you edit Pauline's reagent into the blank sample also,
Yes.
or not?
Yes, I measured the blank falling reagent was also added. You can see here it is written in the blank also in this table itself. Any other doubts?
Yes, ma'am. Yes. Ma'am, what is the highest wavelength which can be generated by UV based spectrophotometer? Like it generates monochromatic light, right? Light or array, we can say.
Actually, yeah, it emits monochromatic light. If we want to take a spectrum, we can take a spectrum at all the wavelengths. And if we want to take at a single point, like we did in this experiment, we can take at single wavelength also.
My question is like, what is the maximum wavelength of monochromatic light it can generate? Like its name is UV visible spectrophotometer. So I
Yeah,
guess
it
it
range
could
also
generate
is
visible
bigger.
light also.
Yes, yes. It can detect at UV range as well as the visible range.
Okay. Ma'am, I have like we have done a Bradford assay for protein estimation. So I just wanted to know like what's the difference between Bradford assay and Fallen slurry this method?
It is a difference in the reagent that we use, and also
Oh,
each test has its own advantages and disadvantages. So there in Bradford, we
yes.
use Comasi dye.
Yes, ma'am. Huh. We did use Kumasi dye.
And anything else? Could you understand the video? If you couldn't, then I have uploaded the slides as well as the video. You can.
I- Yes, ma'am. We were able to see, but it was not. But we can see like if you have uploaded then.
Yeah, you can see that, and recording also you can see both. Shall I move ahead? Any other doubts? Okay. So next part of this experiment is plotting the graph. I hope everyone of you know how to plot a, um, a graph, plot a graph on a graph paper. How to plot a curve on a graph paper. So you have to plot the protein concentration on the x-axis and the absorbance values on the y-axis. And then you have to construct a straight line. And from the absorbance value of the unknown, drop a perpendicular on the x-axis and find the protein concentration for the unknown. So next part is in the Excel. You have to enter the BSA as well as absorbance in two different columns, and then select the chart type scatter and add a trend line and equation and R square value. For this, I will show you one demo. So first you have to select both the columns containing protein concentration and absorbance. And then you have to go into
Ma'am,
insert.
can you zoom a little bit?
What you just asked.
I said that can you please zoom a little bit so that we can see what's
Okay, it is not visible to you.
yes, ma'am.
So you can see this thing in the uploaded presentation. I will explain you here.
Yes, ma'am. Okay, ma'am.
So so first you have to select both the columns, and then in insert you have to go into scatter plot, and then do the right click and then add trend line. And here you have to click on set intercept at 0 and display the equation and then R square value. And from this, you will get in the equation, you will get the slope, and you have to determine the concentration of the protein. You have to divide the absorbance by the slope, and you will get the concentration. So, as anybody has any questions, they can ask.
Ma'am, initially we added solution 1, solution 2, and various other reagents. So, what is the significance of using each of them?
So, the solution 1. So solution 1 and solution 2 basically consist of the copper ions as well as the alkaline solution. Because alkaline solution, in presence of alkaline solution, only copper reacts with the peptide bonds. So it is the condition that is needed. So the first thing is sodium carbonate, which provides the basic medium, and the copper sulfate. And then once we have a peptide and copper complex, we add the following reagent, which reacts with the tyrosine and tryptophan in the protein and gives us the blue purple color for which we take absorbance.
Got it, ma'am.
Any other doubts?
Ma'am, I'm sorry, but I did not understood exactly like how that purity is determined using absorbance, when it was in very earlier slides. Yeah, in this slide.
Purity, actually, the purity is determined by the ratio. Because at a different, in the first thing, 260 by 280, when we take absorbance at 260 nanometer and 280 nanometer and divide the two absorbance value, if the value is 1.8, then it is a pure DNA. If it is less than 1.8, then the denominator value is high. And here 280 nanometer was the maximum 280 nanometer was the absorption maximum of protein. So if there is high protein, then this ratio will be less than 1.8. And if it is 1.8, then it is pure DNA. And if it is more than 1.8, then it is RNA. Means at 260 nanometer, the absorption at 260 nanometer is high. Could you understand? Basically,
Ma'am,
these are the general, yeah. Hello?
Yes, ma'am.
What you were not able to understand in this?
Ma'am, in this in the table which is left to this, we can see like for protein, there is 213 nanometer and 280 nanometers. Ma'am, what is these two?
230 nanometer. In 230 nanometer, basically the absorption is because of the peptide linkage. At 280 nanometer, the absorption is because of the tryptophan and tyrosine residues. And at 260 nanometer, basically the nucleotide bases, nitrogen bases present in the DNA and RNA gives the absorbance. So whichever absorbance is higher, we will know what is present in the given sample. So if your sample contains more of DNA, then your absorbance at 260 will be higher. So your ratio will be the ratio of 260 by 280 will be 1.8 and above.
Okay, ma'am. Okay. Thanks, ma'am.
Any other doubts? Okay, if you people don't have any doubts, start writing the experiment.
Ma'am, what exactly do we have to write? Like this much theory and all this slide you have given us. Like and in last class, ma'am told us like we have to just write two paper and we have to write like with our hands and we don't have to write in Word file or something. So ma'am, in two pages, how much we can write? No more than 100 to 200 words. So what exactly we will have to write? I don't understand.
So you have to write basically first you have to give the aim and then you have to write the principle that we use in this experiment and also the principle for UV visible spectrophotometer. For both you have to write the principle and after that you have to give the materials that are being used which is given in the PPT and then to write the method and also you have to write method in the passive voice in the past tense and the passive voice. That's it. And then you have to give the table.
So ma'am,
The
just this
observation.
table.
Yeah, all the material you will get in the PPT, but it is advisable that you read more about the experiment and include that in the principle. And then you have to
Yes,
plot
ma'am. So
the graph from both Excel as well as the using the graph paper so that you will know through both ways.
yes, ma'am. Yes. So basically, all the things are there in the slides.
Yeah. Any other doubts anyone has?
Ma'am, the submission date for this?
You can submit by tomorrow evening. And I will change I will give you a fresh ID. This ID is actually the old ID that is mentioned in the PPT. I will give you a fresh ID and you can email it onto that. Okay. By
Um,
tomorrow, 5
Ma'am,
p.m. you
but
can.
so, when would you let us know about the ID on which we have to submit it?
Just now I will give you.
Okay, okay,
But
ma'am, sure.
I will write on the Teams.
ma'am, but that we can't write in two pages. Like how many pages in which we have to write? Like all these things we can't write in two pages.
It's not for every experiment 2 pages are only needed. It's not the hard and fast rule. You will use as much as papers you might need to show the practical.
Okay, ma'am. Okay.
Ma'am, what exactly do you mean by tomorrow evening? Like, can you give us a specific time?
5 p.m.
Ma'am, because the thing is, we have a lab tomorrow as well from 2 to 5, and we have
But
classes in the morning. So if you could extend it at least to tomorrow night, then that would be better.
Tomorrow night. Okay, you
Yes,
can give
ma'am,
by 12.
midnight will be better.
Yes, an end of day tomorrow would be better.
Okay, you can give by 12, but that would be the last. No more
Yes.
extensions.
Yeah, okay, ma'am. Thank you, ma'am.
Ma'am, also could you please upload one sample report so that we could just take reference from it?
Uh, okay, but there are, I don't think I have, there is anything I have missed out in explaining. Uh, if you have any doubts, you can ask, but
Ma'am, some of us have to go through the recording once, so like for that, uh, because network isn't that good for everyone.
okay, you can also see the PPT that has been uploaded.
Yes, ma'am. But after that, going through that, we will be able to ask some questions. So we need some time for that.
Okay, okay, you can see you have, I'll give you 1 hour. You can see whatever questions you have. After that, I will take your attendance and ask if you have any doubts. Okay.
Oh, excuse me, ma'am.
Yes.
Is return submission compulsory?
Which
Like, can we
submission?
do it on Word? Like, is it compulsory to write it by hand, or we can use Doc or Word for the same?
You have to write it by hand only. Otherwise, there is. Uh, we won't know now if you have copy pasted or you have written anything. So it's preferable that you write it by hand. At least you'll remember whatever you've written.
Okay, ma'am.
So you can check the PPT that has been uploaded, and if you have any doubts, I will ask at 3:30. If you have any doubts, you can ask at that time, and I will take your attendance at 4 p.m. Okay.
Ma'am, so everybody will have to be in the meeting till 4 p.m.
Did anyone mention?
Ma'am, we can mail our doubts. So like, do we have to stay in the meeting till 4?
Uh, Actually, your practical is for 2 hours. Uh, it's better you ask your doubts in the meeting only. Right?
But
Otherwise,
ma'am, whoever don't have any doubts, so what can we do now?
if you don't have any doubts,
Ma'am,
Yes, ma'am.
yes.
ma'am, you uploaded the PPT at 1 p.m. and we had classes. If you uploaded it earlier, like yesterday, then it would be easy for us in the next experiment. It would be easy for us to ask doubt in the lab because we received the PPT at 1 p.m. and we haven't gone through it yet
But you
due to
can
other.
go through it now also. You have been given time for that. And anyway,
Okay.
I've showed it the whole PPT. So you just have to go through it once again to see if you have any problems.
end the recording so that it gets saved and we can refer to it?
Okay.
Ma'am, can